Hello friends, welcome to the series of lectures of immunology. This is my 17th lecture on the types of immunities. The survival of human species is dependent on its ability to defend itself against the constant threat from microbial infections. Infection refers to the invasion of harmful microorganisms into the body, causing diseases. Infectious diseases cause approximately 25% of all death in the world, but in low-income countries, this can rise to 45%. Microorganisms such as viruses, bacteria, etc. virtually lives everywhere in the environment. They exist in our food, in the water, on our utensil and on our bed sheets. They inhabit the surfaces of living humans and animal bodies and grow abundantly in the mouth and intestinal tracts. But the human body resists, and there is a continuous war between the microbes and the body. In this war, if our body succeeds, the microbes are killed and the person remains healthy without any infectious diseases. If our body fails in the war, microbes invade the internal organs and causes disease. All the organs such as circulatory system, respiratory system, nervous system, excretory system, etc. are the sterile and free from microbes in a healthy human. Microorganisms are kept out of the body by skin, the secretion of mucus, ciliary action, the lavaging action of bactericidal fluids like tears, then gastric acids and microbial antagonism. At every moment, the microbes try to invade the internal organs. If penetration occurs, bacteria are destroyed by the soluble pattern recognition molecules such as lysozymes and complement, as well as by phagocytosis followed by intracellular digestion. Now let's talk about the types of immunities. There are basically 4 types of immunities: humoral immunity, cell-mediated immunity, adaptive immunity and innate immunity. Now let's talk about the first immunity type, that is the humoral immunity. Humoral immunity refers to the resistance developed by the man against microbes by the production of chemical substances called antibodies. Hence, it is called antibody-mediated immunity. Here, the antibodies are produced in the humor, that is body fluids, and hence this immunity is also called humoral immunity. The bacteria, after entry into the human body, releases toxins. The toxins are of 2 types, namely endotoxin and exotoxins. Endotoxins are released as a result of death of gram-negative bacteria. They are lipopolysaccharides. The exotoxins are released by living gram-positive bacteria. They are composed of proteins. Both endotoxin and exotoxins are antigenic and they function as antigens. In response to these antigens, the human body produces antibodies. These antibodies are called antitoxin antibodies. The antitoxin antibodies neutralizes the toxin. The diphtheria and tetanus infections are prevented by the action of antitoxin antibodies. The whole bacteria entering the body are clumped by specific antibodies produced against the bacterium. These antibodies are called antibacterial antibodies. These antibodies attach with the bacteria and the bacteria are clumped and immobilized. The opsonization is a phenomena of coating an antigen with substances such as an antibody or an activated complement component to increase the susceptibility of the antigen to phagocytosis. Opsonin is an antibody which renders bacteria and other cells susceptible to phagocytosis. The antibody which functions as the opsonin belongs to the class of immunoglobulin G. When the bacteria enters the human body, IgG antibodies are produced. These antibodies coat the surface of bacteria. The phagocytes have the receptors for IgG antibodies. The phagocytes migrates towards the antibody coated bacteria and are eaten by the phagocytosis. Bacteria with a capsule are disposed by the method of opsonization. Example: Streptococcus aureus, Bacillus anthrax and Haemophilus influenza. Now, the second type of immunity is a cell-mediated immunity. The resistance against infection produced by the sensitized lymphocyte is called cell-mediated immunity. Here, antibodies are not produced, but specific lymphocytes are triggered against microbes and the microbes are disposed. Cell-mediated immunity gives adequate protection against viruses, fungi, protozoa and certain bacteria. It does not give protection against cocci. When a microbe enters the body, it is processed by the macrophages. While processing, macrophages prepares the cell for immunogenicity and it retains antigen of the microbe on the surface membrane. The macrophage then presents the microbe to the T cell. The T cell recognizes the microbe when a cell-to-cell contact between T cell and antigen macrophage complex is made. This recognition activates a population of T lymphocytes. These activated T cells releases certain chemical substances called lymphokines. The lymphokines mediate the immune response. The lymphokines recruits and draws macrophages to the site of infection and the microbes is destroyed by the macrophages. Many microorganisms lives inside the host cell where humoral antibodies can't reach. Such intracellular microbes are killed by cell-mediated immunity. Viruses replicate only inside cells. Similarly, Leishmania, Trypanosoma cruzi and certain bacteria like Mycobacterium tuberculosis, Mycobacterium leprae, etc. thrives inside the macrophages after their uptake by phagocytosis. These microbes are killed by cell-mediated immunity. The macrophage containing intracellular parasites deposit on their surface some antigenic substances of the parasites. T helper cell, which is a subpopulation of T lymphocyte, recognize the antigen present on the macrophages. These T helper cells bind to the antigen. The T helper cells are triggered and they produce lymphokines. The lymphokines activate the microbicidal mechanism of the macrophage and brings about the death of the intracellular microbe. Now, the third type of immunity is innate immunity. Immunity is a state of protection from infectious disease and it has both a less specific and more specific components. The less specific component, innate immunity, provides the first line of defense against infection. Most components of innate immunity are present before the onset of infections and constitute a set of disease resistance mechanisms that are not specific to a particular pathogen, but that include cellular and molecular component that recognizes classes of molecules peculiar to frequently encountered pathogens. Phagocytic cells such as macrophages and neutrophils, barriers such as skin and variety of antimicrobial compounds synthesized by the host all plays an important role in innate immunity. The innate immune system is also comprised of a soluble factors and cell that detects and responds to infectious agents through binding to relatively non-specific structures like pathogen-associated molecular patterns, also known as PAMP, that are common to many pathogens. Innate responses are initiated through detection of PAMP representing non-self or danger-associated molecular patterns, also known as DAMP, that represents the hidden self. Pattern recognition receptor molecules, which are known as PRR, which can be either soluble or cell-associated, are used by immune system to detect the presence of PAMP or DAMP. PRR engagement leads to a diversity of responses that are aimed at directly killing or engulfing microorganisms via phagocytosis and also results in amplification of immune responses through release of a range of messenger molecules such as cytokines and chemokines. Inflammation is the term used to describe the series of events that surrounds an immune response and includes local swelling due to recruitment of phagocytes and plasma proteins from blood, redness, pain and the temperature elevation. The products of activated mast cell and complement activation collectively promote inflammation. Following the activation of complement with the ensuing attraction and stimulation of neutrophil, the activated phagocytes bind to the C3b coated microbes by their surface C3b receptors and may then ingest them. The influx of polymorphs and the increase in vascular permeability constitute the potent antimicrobial acute inflammatory responses. Inflammation can also be initiated by tissue macrophages that subserve a similar role to the mast cell as signaling by bacterial toxins, C5a or C3b coated bacteria adhering to the surface complement receptors, causing release of neutrophil chemotactic and activating factors. Now, the fourth type of immunity is adaptive immunity. Innate and adaptive immune response are interdependent and cooperate to kill infectious agents. The adaptive immune system is comprised of T and B cell lymphocytes that recognize highly specific structures, that is antigen, on the microorganisms via highly diverse membrane receptors that are generated randomly and are uniquely tailored to individual pathogens. Adaptive immune responses exhibits 4 immunologic attributes. They are specificity, diversity, memory, and self/non-self recognition. The high degree of specificity in adaptive immunity arises from the activities of molecules like antibodies and T cell receptors that recognize and bind specific antigens. Antibodies recognize and interacts directly with antigen. T cell receptors recognize only antigen that is combined with either class 1 or class 2 major histocompatibility complex, that is MHC molecules. The two major subpopulation of T lymphocytes are CD4 T helper cells and CD8 T cytotoxic cells. T helper cells secrete cytokines that regulates immune response upon recognizing antigen combined with the class 2 MHC, whereas T cytotoxic cell recognizes antigen combined with the class 1 MHC and give rise to cytotoxic T cells which display cytotoxic ability. Exogenous or extracellular antigens are internalized and degraded by antigen presenting cell that can be macrophages, B cells and dendritic cells. The resulting antigenic peptide complexed with class 2 MHC molecules are then displayed on the cell surface. Endogenous or intracellular antigens, example viral or tumor protein produced in altered self cells, are degraded in the cytoplasm and then displayed with class 1 MHC molecules on the cell surface.